All right, so today we'll be talking about the primarily the origin of the vertebrates, but the chapter deals with the beginning of the vertebrates up through just before the time the vertebrates moved to land as amphibians. So vertebrates began as fish, and about 100 million years ago, after they'd moved to land, there was three dominant vertebrate classes: fish, reptiles, and birds. And this just shows how a scene that might have appeared 100 million years ago. There were modern fish and crocodiles and birds at that time. And then shortly afterward, if you want to call 35 million years shortly, the mammals took over. And then 65 million years later, we showed up uh, just in the last, million years or so or less. So anyway, today we'll focus more on the origin of the vertebrates. And so here's the news. All modern vertebrates, including humans, descended from a vertebrate fish that lived at least 518 million years ago. And so our ancestor at least resembled this vertebrate fish discovered in the 518 million year old Chenjiang Lagerstätte. It's a famous fossil formation. And you might be wondering, how did I, a person who didn't necessarily think that he was descended from a fish, learn to deal with the fact that my great-great-great-grandmother was a fish? Well, in my case, I'd been trained through a series of disappointments about my ancestry, and it began when I was a child. I don't know why, but when I was growing up, I had a dislike of French people. And not that I actually knew any French people, but I just disliked France and the whole concept of French people. And then I was told, and I quote, that there was a bad Frenchman from Louisiana in our family tree. And I made this picture in Mid-Journey bot, Imagine AI. I just typed in bad Frenchman from Louisiana. Well, I was very disappointed to learn that I had a French person in my ancestry, but things got worse when I was an adult. As I was studying fossils of Homo species, I came to the conclusion that I was descended from apes. And I actually felt nauseous and sick to my stomach when I realized this. But after these disappointments, it was no big deal to learn that I was descended from a fish. And I look at it like we were robbed like 3 times. And the first 2 times I was extremely angry and paranoid about being robbed. But the third time, it really didn't matter. It was just like, oh, well, I got robbed again. So I think after the first 2 times with my ancestry disappointments, I felt bad, but I'd become used to it. So it didn't really matter. And actually, as I study this fish, I've actually become very excited that I'm descended from this fish because it had all the genetic architecture needed for the eventual evolution and appearance of humans. So this chapter begins with the concept of homology. And when evaluating the sequence and connections of organisms in the fossil record, scientists use the concept of homology. And this concept originated in the early 19th century with Geoffrey St. Hilaire. And then Darwin extended this concept by identifying homologous structures as a basis for defining evolutionary relationships. So scientists establish homology through conceivable evolutionary processes or through observed transition in the fossil record. So they're able to connect one organism to another based on their similarities. Section 9-3 focuses on non-homologous characteristics between the first vertebrate in the fossil record and previous organisms in the fossil record, of which there are many. And in this presentation, I think I've only included 8, but there's a few dozen others. And section 9-4 focuses on the formation and characteristics of the vertebrate nervous system. So scientists attribute almost all of the advanced vertebrate features to the vertebrate neural crest, which you might have heard about. It's the origin of stem cells. So neural crest cells form placos and vesicles that then form the sensory organs based on signals from the neural crest. And the neural crest forms many unique vertebrate organs, such as the kidney and chambered heart. Likewise, the cartilage and head are formed by neural crest cells. Then we'll look at section 9-5, which focuses on the neural and body patterning genes in vertebrates. And although we have similar genes as in vertebrates, they were amplified in the vertebrates. Section 9-6 describes the long, slow evolution of fish in the Paleozoic era. Beginning with jawless fish, evolution proceeded with fish with jaws, cartilaginous fish such as sharks, bony fish, and then two divisions of bony fish which still exist today, ray-finned fish and lobe-finned fish. And then section 9-7 reviews a date by leading paleontologists about vertebrate origins. So vertebrates are within the Deuterostome phylum, which is classified based on embryo development. And the word deuterostome means second mouth, which means the mouth forms in a second opening. And within the Deuterostome phylum, we are within the chordate class. By we, I mean vertebrates, which means that we have a notochord, dorsal nerve cord, postanal tail, and pharyngeal slits. And one of the great mysteries of animal evolution is that vertebrates are sort of upside down from other phyla, invertebrate phyla. So in comparison with annelid worms and other invertebrates, our dorsal tube is on the top. Our dorsal nerve tube is on the top, which is called the dorsal side, and the annelid worm nerve cord is on the bottom, which is called the ventral side. And then both we and the other triploblastic bilaterian invertebrates have a central gut, which is the yellow circle in the middle. So if we're looking for homologous characteristics, we'd be looking for an organism that has a similar structure to ours, meaning the dorsal nerve cord and the ventral gut. I have that slightly wrong. Anyway, let's just say dorsal nerve cord, central gut. So you might remember the fossil of Pikaia from the Burgess Shale from the last lecture. This is that little primitive fish. And an artistic reconstruction of Pikaia is shown here in line E. Actually, it's line F. Shown here in line F. And this is from a paper by Mussini et al. From 2024. And I noticed it's also quoted in Wikipedia. So I think it's a pretty influential paper at this point, even though it just came out. There's been major controversy about the inclusion of Pikaia in our family tree, but Mussini et al. Argued for it. And one of the major roadblocks to Picaia's status as a vertebrate ancestor was the position of the nerve cord in the gut, which were thought to be in the opposite position from the vertebrates. But Mussini et al. Reinterpreted Pikaia as having a dorsal nerve cord instead of a ventral, which means on top of the organism in a ventral gut. So in this image, they show the similarity of Pikaia shown here in line F with another Cambrian chordate called Unanozoan. And so what they did is they, like we talked about before, they compared the homology, homologous structures. You can see that these two organisms look pretty similar. So they would be called homologous. And also, Pikaia has a similar muscle structure and swims with a side-to-side motion as with vertebrates. So that'd be another thing they would be looking for. It has this chevron-shaped muscle structure. And although it did have the side-to-side swimming motion, it was probably more like an eel rather than what you think about with a fish. And the reason for that is I don't think it had a notochord, which would be a strong cord running down the body. It had a nerve cord, but not a notochord. And in contrast, annelid worms swim with a up and down motion. So based on their interpretation of the similarity of Picaia with Unanozoan, Mussini et al. Envisioned this path to vertebrates along the chordates. So you can see here's Picaia circled in red. And you can also see a Unanozoan here, actually before Picaia. And I haven't really mentioned it, but some people put sea squirts in the line of evolution of the vertebrates. Sea squirts are those like the tunic, they're also called tunicus. They have this tunic around them. They swim for a short little time, like in their larval stage, and then they bury their heads in the sand and grow these tunics. But because they have fish prior to the vertebrates, they put the tunicate or sea squirt kind of as a cousin to the vertebrates rather than as ancestral to the vertebrates. And this slide just lists the names of these organisms. So a lot of scientists think that vertebrates or deuterostomes began with ambulocraria, which are also called hemichordates. They're sort of like tubes, and I think the animal's inside. And then next they put vetulicolians, which would be these kind of shell-shaped things, but with a fin on the back. And they could swim. So those would be the first swimming organisms. They're very small. They're all showed kind of the same size here, but they aren't necessarily. Then Mussini et al. Put Unanozoan and then Picaia. Then they put Amphioxus here. Amphioxus looks awfully similar to Picaia. And what an Amphioxus is it is a modern organism, but it's also very similar to the shape of a larval lamprey, which is why scientists put lampreys at the base of vertebrate evolution. But they just had a pretty major study come out from University of Chicago, major paleontology lab, where they found evidence of lampreys about 200 million years after the Cambrian with no larval stage. So that paper, they said, well, we don't think vertebrates descended from an Amphioxus-like organism because they didn't even exist. There was no larval stage in vertebrates back in the Cambrian, or in lampreys anyway. So anyway, there's arguments on both sides. Papers are coming out. A person named Malat, who kind of came up with the Amphioxus idea, just came out with a paper saying, oh, not so fast. We shouldn't take Amphioxus out of the vertebrate origins. So it's still controversial. But anyway, my opinion has been also, I agree with that Lamprey paper. Not that my opinion matters, you know, as far as that goes, but I think that the paper is pretty convincing. Okay, so that's the path to the vertebrates. The thing to know about Picaia is it's an invertebrate without any of the vertebrate characteristics, which I'll show you in a little bit. Okay, so here we have a progression from Picaia to Metaspragina, which was an organism similar to Hycoichthys. It was a fish in the Burgess Shale about the same time as Picaia. Both of them appeared about 510 million years ago. And then we have the progression after the fish. We have crocodiles and birds, which appeared about 100 million years ago, and then humans. And it would seem that there's a larger difference between these guys and these guys, or girls, than between each other. But actually, there's a much bigger difference between Metaspragina and Picaia, so between a vertebrate and an invertebrate, than there is between the early fish and the later evolved vertebrates. So what I'm going to do now is I'm going to go over the differences between Metasprigina and Pikaia, the vertebrates and the invertebrates. So even though they look similar, their characteristics are way different. Okay. And so Henry G. is a senior editor of Nature Journal, and he's considered one of the world's leading experts on vertebrate origins. And in 2018, he wrote a book called Across the Bridge, Understanding the Origin of Vertebrates. And this is a quote from the book. At first sight, many of the characteristics features of the vertebrates appear to have evolved all at once. This explains why vertebrates appear so different from anything else in the animal world. However, it is legitimate to ask whether the apparently unique features of vertebrates evolved not simultaneously, but one at a time, and if so, in which order, and whether some of them might be found, even if in some more modest form, among invertebrates. And he's actually the one who proposed that vertebrates might be descended from Vetulicolians, which was in that family tree of vertebrates that I showed you earlier. So in summary, he stated that there's no known evolutionary pathway to the vertebrate characteristics in the Chenjiang Lagerstätte or Haikuictus. So he's saying it is as if they appeared all at once in Haikuictus. So now let's look at the non-homologous vertebrate characteristics. Non-homologous means that there's no evolutionary pathway to these vertebrate characteristics. And these are just some of the novel characteristics in vertebrates that I'll be showing in this presentation that are not found in invertebrates. For example, there's no example of a primitive vertebrate type of eye in the fossil record prior to the first vertebrate in the fossil record. There are eyes in cephalopods or squids, but those were like 200 million years after this. Okay, so the first thing we'll talk about is the neural crest stem cells. And the neural crest is a sort of tube that forms in the upper or dorsal side of the vertebrates. And so it's the major embryonic structure that defines vertebrates. And neural crest stem cells, you've probably heard of stem cells. They form in the neural crest, and then they migrate out to different locations in the embryo, and they form the different organs and structures, such as the cranium and face, the brain, the four parts of the central nervous system. The neural crest and neural crest cells are not found in invertebrates, such as Pikaia or any other invertebrates. And although Pikaia had actually, I think I'm wrong about that. I don't think it had a notochord like vertebrates. It had a nerve cord, but it didn't have a neural crest. And neither do sea squirts or tunicates. So as I'll show you, the neural crest is like an add-on that triggers the formation of vertebrate characteristics. Okay. So one of the most amazing structures in vertebrates, and even in the original vertebrates, are cranial sensory ganglia. And they're clusters of neurons that connect the eyes, ears, and other sensory organs to the brain. And they originate from neuron clusters in the ectoderm. So the ectoderm is like the skin, which become the placodes where the eyes and ears developed. And cranial neural crest cells shape the sensory ganglia structures. So these structures here, so it has to go through the cranium out to the senses. And there's more here in humans than there would have been in the first fish, but these structures did exist in the first fish. And so some of these structures go through holes in the cranium, and others go along like canals that are made. So like our eye as a whole. And so researchers found structures in Haikuictus that represented eyes, ears, and a nose. So these would have been connected to the brain by cranial sensory structures. Okay, the next non-homologous characteristic is the vertebrate 2 or 4-chambered heart. It's unique. and not homologous with invertebrates. There are many non-homologous features of the vertebrate heart. And these features are formed by the vertebrate neural crest. One of the unique features of vertebrates is the kidney and the heart-kidney system. So the heart pushes blood through the kidney, and the kidney cleans the blood. And there's no fossil evidence of a kidney in Hycoichthys, although Dittrich thinks that there was a kidney there. And the kidney was essential for vertebrates to migrate to land, to deal with I don't know exactly what, but in order to become large, we needed a kidney to clean up our blood. The early Cambrian vertebrates had an internal skeleton and cranium. And the vertebrate internal skeleton and hydroxyapatite from which it is made are vertebrate novelties. So there's no invertebrates with an internal skeleton, nor bones constructed from hydroxyapatite, which is, I think their bones are made from calcium carbonate. And also there's still no invertebrates with a head or cranium, which we also have and which was in the initial vertebrate fish. So vertebrates have image-forming eyes, and Hycoichthys and Mylocunmungia, those early vertebrate fish, had vertebrate eyes. And there's no observed transition to the vertebrate image-forming eye in the fossil record. There's some proposals, but there's no evidence of an eye in any other organisms back then. And the shape-shifting vertebrate lens allows it to focus on near and far objects, and the adjustable pupil allows the eye to adjust to different levels of light. The choroid here provides oxygen and nutrients to the retina. So here's the retina. And the sclera, the white of the eye protects the eye. The ciliary body and attached muscles control the shape of the lens, so that allows us to focus. And the attached skeletal muscles also change the direction of the eye. And so light passes through the transparent nerve layers back to the rods and cones, which are at the back of the retina, and then the signals from the rods and cones then move through nerve fibers to the front of the retina, where they eventually reach the ganglion cells. Remember, I talked about the ganglia that connect the brain to the senses. And then the nerves in the retina process information, identify shape and bright points of light prior to passing the information back to the brain. And so squids have eyes, and they're cephalopods, and they resemble vertebrate eyes in outward appearance, but they do not have all these capabilities to move and focus. And they also evolved like 200 million years after the vertebrate eye. Okay, there's fossil evidence of otic structures in Hycoichthys. And an otic structure is like the bones in the ear that, capture sound and allow sound to be processed as, pressure waves and so on. Some insects do have hearing, but there's no evidence which would be otic structures, but there's no evidence of otic structures in Cambrian invertebrates. And I just wanted to summarize what they found in the fossil Hycoichthys by some leading paleontologists. So in 2003, Shu said this: Hycoichthys shows significant difference from other fossil agnathons or jawless fish. Key features include a small lobate extension to the head with eyes and possible nasal sacs, which would be the sense of smell, as well as what may be otic capsules for hearing. A notochord with separate vertebral elements is also identifiable. And phylogenetic analysis indicates that this fish lies within the stem group Craniates. And in 2014, Morris and Karen described Metasprigina. Remember, that's that other fish I showed you in some of the slides. This primate fish displays unambiguous vertebrate features, a notochord, a pair of prominent camera-type eyes, paired nasal sacs, possible cranium and arcualia, W-shaped myomeres, which is the chevron-shaped muscles, and a post-anal tail. So in summary, the fossils indicate that Hycoichthys and Metasprigina had the senses of sight, smell, and hearing. And although invertebrates have or had chemosensitive cells, the vertebrate sense of smell has an entirely different level of complexity. And it's based on olfactory receptor neurons. So here's the olfactory nerves. And they extend tiny hairs into the mucic in the olfactory epithelium, which captures odors. And the combination of different receptors binding to the odorants in different ways leads to the sense of smell. And then receptors are joined together in glomerules at the olfactory bulb. So here's the olfactory bulb. Then it goes back to the brain. Okay, so this shows obviously the human nervous system, but the basic components in the there's 4 different parts to the nervous system. There's the peripheral nervous system, enteric nervous system, and central nervous system. Paleontologists think those were in place in Hycoichthys, according to Henry G. Remember, he's that famous paleontologist. And all vertebrates have olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, auditory, glossopharyngeal, vagus, accessory, and hypoglossal nerves. But invertebrates do not have these nerves. Another important part of our physiology is the pituitary gland, which is behind the nose, and it controls appetite, sexual arousal, and many other glands. And it also regulates reproduction, growth, and stress. And according to Henry G., the pituitary gland and other endocrine glands were in Hycoichthys. The Hycoichthys brain was not extremely large in comparison to invertebrates at the same time. It was only about 2.5 centimeters long, but it did have a tripartite brain structure, which is the same as modern vertebrates. So paleontologists consider it to be complex. But early Cambrian invertebrates also had fairly complex brains because they had to control, the sensory signals from whatever types of sensors they had and control the organism's movement. Okay, so that's all the non-homologous features I'm going to go over today. But so now we'll move on to the section called neurulation. And it's the process of the formation of the neural tube. So here's the neural crest, here's the neural tube, and here's the neural crest in the next phase. So this is all going on in the embryo. Here's the ectoderm, which is like the skin. And here, so here's the epidermis, the skin, and then the neural tube, and the ganglion going to the spine. And anyway, this is what happens in embryos. These things form. And then the neural crest then breaks up and forms individual stem cells. And then the stem cells in the neural crest migrate to different parts of the body and produce most of the unique vertebrate nervous system and other organs. So although many of the genes in the neural crest are related or even the same as genes in other invertebrate organisms. None of the genes that direct the movement of neural crest cells are present in invertebrates. And the system of neural crest cells migrating to locations in the body and forming nerves and organs is a novelty of the vertebrates. Okay, and you can read more about that in section 9-4. All right, let's move on to section 9-5. Believe it or not, I actually drew this picture. But what you can see is there are similar genes. These are all gene names. There are similar genes in vertebrates and in similar positions in vertebrates and annelid worms. So like green is twist and paraxis. We've got that in annelid worms, and up here, and I'm sure I'm saying these names terribly. Brown is bra, foxe, and twist, and so on. I'm not going to go through it all, but the locations of genes are very similar in annelid worms and vertebrates. You know, the structure, their arrangement. So there's an obvious ancestral connection somewhere between annelid worms and vertebrates. Okay, one of the most important, if you want to call, you know, every gene's important, but one of the most important genes in invertebrates and vertebrates is the Hox genes. They're arranged in a cluster. For example, here's the fly, and I think the fly has 14 of these genes all arranged in one cluster, and they're arranged in the same pattern as they're expressed in the organism. For example, the blue genes at the back are at the back of the fly. So they're sending out signals to the organism to make certain structures in certain parts of the body. And then this light blue is next in the fly. Then we've got the yellow, then the orange, then the red, and then I don't really see the purple here, but anyway. So we have a similar set of Hox genes, but we have 4 clusters, and that allows for the increased complexity in vertebrates. But you can see, again, we've got the same clusters being expressed in the same order in the vertebrates and the invertebrates. Okay. And you can read more about all of these genes in section 9-5. Then section 9-6 describes vertebrate fish evolution. I have lots of nice pictures of different kinds of fish. But we start out in the Cambrian with jawless fish. So those would be the Hycoichthys and Mylocunmungia and other jawless fish. Then eventually fish developed jaws. So it took like 100 million years to develop jaws. And those would be called the placoderms. And so that happened in the Devonian. And then came sharks, which are cartilaginous fish. So you can see the sharks are still around. The placoderms were not as competitive as the cartilaginous fish. So they were, even though they were huge, huge placoderms, like I can't remember if it's 10 feet long or 10 meters long. But anyway, we had cartilaginous fish, then we had something called spiny sharks. Then we had the 2 types of bony fish. We had ray fin fish and lobe fin fishes. And actually, all land animals, birds, reptiles, and so on, they're descended from lobe fin fishes. So we'll go over that next time. The evolution of amphibians from lobe fin fishes and so on. Okay, but this section is very long with lots of good pictures, not just this one graph. Then there was a very interesting debate recently by leading paleontologists, a lot of the people mentioned in the chapter, or at least some of them. And so they had a debate about the fossil record prior to vertebrates. And it's a very well done debate. And so the molecular clock D&E people think that with the differences between vertebrates and invertebrates, there must have been a long period where they diverged a long time ago, or a long time before the Cambrian. And their initial estimates was like a billion years ago, they started to diverge. But then the Cambrian's 520 million years ago. So that would leave about 500 million years. And then they whittled that down to like 600 or 700 million years ago. That the divergence between invertebrates and vertebrates happened. But the difficulty with that is that most paleontologists who deal with the Ediacaran formations think that actually animals began to evolve with the Ediacaran biota and the sea anemones, and then the phases of this progression that I showed you last time, where the first even crown, meaning ancestors of the like annelid worms and arthropods appeared like 535 million years ago. And then the organisms such as Picaya and so on, they wouldn't have appeared much longer before the Cambrian. So the question is, how did this all happen so fast? And so the debate deals with these paleontologists and molecular clock people, but they're all like paleontologists, saying, well, is this really true? Did the animals evolve in such a short time before the Cambrian? Or is the evolution of the animals hidden and we haven't seen the evolution of the vertebrates? They're there, but they're just not showing up in the fossil record. And when did they start evolving? So this debate is very interesting. I have the YouTube connection here. Actually, I'm showing it on 2 lines, but it's one address. And so you might be interested in that debate. All right. So obviously, there's a lot of philosophical and religious perspectives on the origin of the vertebrates, or just the origin of animals in general. But I would say that theistic evolutionists and atheists would just say that they evolved naturally. Progressive creationists, I don't think they really focus in on origin of the vertebrates as some seminal creation event. At least I haven't heard that. They would just say that maybe different organisms had to be created at different times. And then young Earth creationists would say that, God created every species as it is. Like, we're not descended from fish, especially. Okay. So here's Guyot. So Guyot had the invertebrate animals in Silurian rocks, verse 20, and that was last lecture. And then he had fishes in Devonian rocks as the meaning of Genesis 1:21. Well, now with the fossil record, we move everything back about 100 million years, But it's the same sequence as today with invertebrates and then vertebrates. And the reason he said that is so God created the great creatures in the sea and every living thing that which the water teems, and that moves about in it according to their kinds, and every winged bird according to its kind, and God saw that it was good. And this actually, the word is moving thing, and it actually can refer to the movement of a fish. So all these things, the great creatures of the sea would be fish, or I think it's crocodiles. The only other time that the word is used, it refers to crocodiles in the Bible. And then the other things in this description are vertebrates. So that's why he aligned verse 20, which the water swarms with the invertebrates, and then verse 21 with the vertebrates. And he thought, Guyot thought that God created every, like plants, the invertebrates, the vertebrates. But I personally don't really think it says that. I think it says that God created the vertebrates, and it says that the seas naturally formed the invertebrates. So I think that the vertebrates, since they're so non-homologous and there's such innovation, I think that to say that they were created is a reasonable thing to do. All right, we'll see you next time.